Thank you so much for joining us. Welcome to the webinar Availability of and Accessibility to Biologics, Implications for COVID and Beyond. This webinar is being organized by the People's Health Movement and Third World Network. Today with us we have Professor Schellekens and Satyajit. I'll be introducing them in a while. We will wait for participants to join us. It usually takes a few minutes for the participants to join us. So we'll just wait for a few minutes and then we'll directly start the webinar. So thank you so much. So we are having around 40 to 45 attendees right now, and we will start in a while. So excuse us for a bit of delay there. So thank you so much, and we'll now start. We have around 48 attendees. So welcome to the session and the webinar. This webinar is actually to discuss the accessibility of biologics and what is the scope of biologics in actually dealing in the response with COVID as well as other diseases. Because right from March, there has been a lot of speculations also, and there has been a lot of hope that we will be having a lot of positive response from the side of big molecules that we say in the biologics, antibody treatments, and so on. Because it's also said there is a kind of understanding in lay sense that before vaccines comes, actually the main focus and the main response could be in the form of biologics and so on. So today joining with us in this webinar are Professor Schellekens. Professor Schellekens teaches pharmaceutical biotechnology at the University of Utrecht. He has been involved in making, changing the standardizations required for biosimilars and has been an advocate of access to biosimilars and biologicals from quite some time. And he has also been instrumental in helping local production of biosimilars in different places. And our second panelist today, we have Professor Satyajit Rath. Professor Satyajit Rath is an adjunct faculty now at the Indian Institute of Science Education Research. And he has been associated with the People's Health Movement and is a known science activist in India. And sadly, today Chaitali Rao, who is the third speaker, could not join us today due to ill health, and conveys her apologies for not coming. And we will be starting with our session first with Professor Schellekens, who will be introducing biologics, because as they say, it takes quite some time to actually understand what they are. And I hope that we'll have an enriching discussion. And Professor Satyajit would be acting as a discussant here and sharing, also having a discussion. And we will request the attendees to kindly type their questions in the Q&A section, and the chat function is mostly for sharing any information and so on. So I'll now give it to Professor Schellekens and request everyone else to mute their video as well as audio. Thank you so much, Professor.
Okay, I first want to thank the organizers for inviting me today. A little bit more about my background. I'm in fact, I'm not a pharmacist, I'm a medical doctor, but I've been involved now for more than 40 years in looking at the safety of biologics, and also active trying to make biologics affordable in third world countries. This will be the content of my webinar today. I will explain to you what a biological drug is and what makes a biological drug different from the standard small molecules. I was also asked to explain what we know now about the use of biological drugs in COVID-19, and then I will give you my ideas on how we can increase the access to biological drugs. So what is a biological drug? There are a number of definitions. The Europeans define biological drugs by the way they are produced. So if a drug is produced in or derived from a living cell, then you are a biological drug and you have to fit all the regulations for biologics. The Americans are a bit more pragmatic, and they define biological drugs mainly by the structure. So biological drugs should have proteins in it, or nucleic acids, or both. There are, if you look at examples of biologics, it's really a mixed bag. You have the proteins like the interferons and the insulins, the poietins, and of course the monoclonal antibodies, the most important biologics in this moment in time. Nucleic acids in the form of gene therapy factors and siRNA, vaccines, which of course are important in the discussion on what to do about COVID-19, cells, etc. But in general, if we talk now on biological drugs or biopharmaceuticals or whatever you call them, we are talking about mainly proteins, and the majority of the products currently on the market are monoclonal antibodies. So what's the difference between a conventional drug, a small molecule like aspirin, and biologics? Well, the different size. Biologics are often very large, they're very complex, they're unstable in most cases. You are able to modify proteins much more than you can modify a classical small molecule. They differ, as I already told you, in the way they are produced. Chemicals are made by chemistry, and in biologics are made mainly in living cells. that has issues if you want to make copies, but I will discuss that later. Well, a small molecule in principle is easy to characterize, but a biologic you have a difficulty, and the main difficulty, and for me the main difference between a small molecule and a biologic is the heterogeneity of a biologic. A biological product is always a mixture of related molecules. To give you an example, if you look at a monoclonal antibody, there are a potential 1 billion different related molecules that can be present in the preparation. Another difference is, and that's is still one of my main scientific interests, is that biologics induce antibodies in patients. Not all patients, and the level of immunogenicity differs between biologics, but they are immunogenic, and that can have clinical consequences. While in most cases, small molecules do not induce an immune response. What's the importance of biologics? I mean, the upsurge for biologics came 40 years ago with the introduction of recombinant DNA technology and hybridoma technology that makes it possible to produce any biologic in any amount in cells. In 1983, we had the first biologic introduced in the form of insulin, human insulin, but now we have hundreds of biologics, and they opened up possibilities to treat diseases that we were not able to treat before. Another important issue with biologics, and that's important for the safety, is that they have a very high degree of specificity, and often they have also a prolonged half-life, which means that they are you don't have to dose them every day or a couple of times a day. You even have products now that you can you only need to treat the patient once a year, and in general, biologics are really safe drugs. Another issue I will discuss is that, well, the first products came in '83. In the last, let's say, 15 years, many data protection and patents have expired on biologics, and that opens up the possibility to introduce copies of, we call them biosimilars, which are less costly than the originals. What are the drawbacks? I think the most important drawback is the price. Biologics are among the most expensive drugs. I think the world record now is the Zolgensma, the gene therapy vector from Novartis that costs more than 2 million for a treatment. The access to drugs is limited in many parts of the world, and what we are seeing now, that is more and more including also the richest parts of the world, the industrialized world. To give you an example, even in the European Union, 50% of the patients with orphan diseases for which there is a treatment cannot afford or have no access to these drugs because of the price. For biologic, often you need experience and also advanced diagnostic facilities. That may be a problem in third world countries. They are normally injected or given by transfusion. That means that in some cases, the patient has to go to a hospital once or twice a month to spend half a day there to get a transfusion. And of course, because of their stability issues, you need a cold chain when you manufacture the products to the bedside, which can also complicate the use of these products in third world countries. Now let's go to drug pricing, which is, I think, the most important problem we have now in the world of medicinal products. Well, you know, there are different opinions about what drives the cost of the drugs. According to industry, it is the cost of innovation. But if you look at it in reality, then it's caused by monopolies, and it is a typical monopoly pricing. And in contrast to what people think, most people will think, or the industry is claiming, high drug prices do not lead to innovation, but they lead to reduced innovation and an increasing emphasis on a limited number of diseases, and that does not include the neglected diseases of the third world. Well, I think you have to look at the drug pricing in the general context of what we think is a crisis in the pharmaceutical world, because what we see a declining number of drugs, most of the drugs are Me Too's, increase in prices, increase in orphan drugs, that means drugs for limited populations, because they are orphan drugs. They don't need a full dossier, so you have seen more and more drugs of which there are insufficient data available, especially clinical data, which makes it impossible for reimbursers to really study the cost efficiency of these drugs. Well, I told you there is a decline in the indications for which new drugs are introduced, and what we see mainly now is anti-cancer drugs and drugs for chronic diseases. And another phenomenon we see both in the US and in Europe, we see an increased number of drugs which have no efficacy at all. Well, this is to show you that this is not sustainable. And what we see here, I will not go through this complete slide, but what we see is that we predict that the days of big pharma are over, and that we need to look for alternatives. Well, drug pricing, there are also discussions on is this drug pricing increase real and whatever. I think this is the best example I could find on showing the world that we are on the wrong track. And this is a relatively simple study by Sloan Kettering in New York. And what they did is look at the cost of 1 month survival in cancer in the last 40 years. And you see that in 1970, that was about $100, and that is now, it's a logarithmic scale, by the way, otherwise it would fall off the screen. But now we are approaching the $100,000 for 1 month survival. We see a tenfold increase every 10 years. So that means that in 10 years from now, we are expected to pay 1 million for 1 month of cancer survival. Well, this is a paper which explains why high prices lead to less innovation. I would suggest that you try to find this paper and read it, because it's very convincing that high pricing stifles innovation. Well, what about the expiration of patents of biologics and the possibility of biosimilars. Are these biosimilars, as we have them now, on the market as a solution? Well, first, I think we need to emphasize that the copies of biologics, the biosimilars, have to follow a very specific pathway. They cannot follow the generic pathway, but in Europe, as first jurisdiction and later in other countries, specific regulatory pathways were introduced, and the arguments were always that the clinical performance of a biotherapeutic is dependent on the manufacturing process. And because of the manufacturing process differing between the company making the copy, the biosimilar, you need clinical studies to be sure that the drug is safe and has a clinical effect. And also to notice that even if there is a small difference in manufacturing process, that may influence the clinical effects and the pharmacodynamics and safety of a biotherapeutic product. So this approach was developed for developing a biosimilar. It's the McGamish pyramid. It shows you that you start with physical, chemical, and biological characterization of your copy in the laboratory. You compare it with the original. You try to make it as similar as possible. And if you've convinced yourself that there are no major differences between your copy and your original, you move on to preclinical studies, PK/PD, and in the end to clinical trials. So what you do is you develop a similar product in the laboratory, and the clinical phase is not to show similarity, but to confirm similarity. This slide says the same thing, but it is in writing, and as you can see in the last bullet point, the paradigm is also that the similar the product is in the characterization in the laboratory, the lesser the extent of clinical trials and the clinical data you need to convince the authorities that your product is in fact a biosimilar. Was pioneered in Europe. The first guidelines were in Europe introduced in 2006. And you see above the line the number of biosimilars that we have currently in Europe. Below the line, you see the FDA approvals, and you see that they were a bit lagging behind because they didn't have a regulatory framework yet in place. But through Obamacare, the Affordable Drug Act, there was also a regulatory pathway for biosimilars in US, and we see an increasing number of biosimilars also introduced in the US. Well, have biosimilars, sorry about the misspelling, solve the problem for the third world countries? I don't think so. If we look at the biosimilar pricing in Europe, we see that you see about, in general, sometimes it's more, but in general you see a 20 or 30% rebate compared with the original product. But in my view, that is still too expensive for lower middle income countries. And then it's the question, why? What should we do? Why are they still too expensive for lower middle income countries? Is it the complexity of the products? and their production? Is it the development costs, or is it the complexity of the regulations? Okay, what are the main lessons from the development of the biosimilars? First, the biosimilars have a high level of heterogeneity. I think that was more or less became more general knowledge because of the biosimilar introduction. What it also showed, because all these manufacturers were looking at different batches of the original products, and what they saw was sometimes very big batch-to-batch variation. And the characteristics of a biologic changes over time. And also that in general, because of the development of the biologics, but also the biosimilars, the many products that we have on the market, it also showed that the biopharmaceuticals are in general very safe drugs, which also gives you, I think, more degrees of freedom in developing a biosimilar. Well, this is an example of differences found in different batches of the reference product during a biosimilar development. There are now many more papers on this topic, but I show you one paper from Korea looking at, I think in this case it was Herceptin, and looking at the differences in glycosylation. And if you study these slides carefully, you see that during the time there are quite some drifts in glycosylation patterns. By the way, also biological tests were done on the different batches, and they also showed differences in biological activity in vitro, so in the lab. Doesn't mean that there were any clinical differences, by the way, between these batches of Herceptin. Well, what are the safety issues of biologics? I think it's very important to realize that a biologic has no toxicity because it doesn't enter cells, doesn't interfere with the internal metabolism, and of course it has side effects, but the side effects are always the pharmacodynamic effects, the effects of the product itself. It's immunogenicity, but that's nowadays something you can well control in biosimilars. And of course, with any injection, you see a lot of skin reactions, but we know that the skin reactions in 99% of the cases have nothing to do with the original product, but are formulation dependent on the excipients. So what is, the discussion is, biosimilars are still too expensive for third world countries. So we have to do something about the development requirements, and especially we have a discussion now on do you need clinical trials, because the clinical trials make up, I think, most of the costs of the biosimilar development, and that also makes it impossible for many manufacturers in third world countries that are in principle are well equipped to make a good biosimilar from a technical point of view, but don't have the funding to do a 40 or 50 million euro clinical trial. So, and there is an increased realization also among regulators in Europe and national regulators that we are starting to doubt whether we need clinical trials in the vast majority of cases of the biosimilar development. And if you look at the regulation, it is also quite confusing. And there is a lot of contradiction in what the regulators say about the clinical trials and the need for clinical trials. Let's take the statements in the European regulations as an example, but you can find them in the WHO regulation and also in the FDA regulation. So they say the smaller difference can have a major impact on safety and efficacy. They also say there will always be difference between a biosimilar and the reference product. The comparability and quality between the biosimilar and the original is a prerequisite for reduction of clinical trial data, but they say at the same time that clinical data cannot be used to justify significant differences between biosimilar and the reference products. And these statements do not add up. If there are always differences, and these differences can have always clinical effects, we need clinical data in all cases, and even comprehensive the clinical data will not be helpful in justifying these differences. And in the end, if you follow these rules to the letter, no product can ever be accepted as a biosimilar. So what are clinical relevant differences in my view? All these differences we have seen between biosimilars and the originals and between batches of the originals and biosimilars. They are not relevant, but because if we look at the clinical effects and the safety issues of the biosimilars, we still are waiting in Europe for the first side effect reported that was really associated by the biosimilar itself. Of course, biosimilar side effects are reported, but they are the same side effects as the side effect of the original product. The data show sometimes that there are differences between batches of original products, and sometimes these are much larger than any regulatory authority would accept between a biosimilar and the original. And in my view, there are only two critical differences. One is aggregation, the level of purity, because it leads to differences in immunogenicity. So if you show me that your biosimilar is as pure as the original, there will be no difference in immunogenicity, and the other is potency. I explained to you the side effects are always the pharmacodynamic effect of the product, and that means that it is 100% associated with potency. So if your potency has this, your product has the same potency as the original, it will have the same side effect profile. Also look at the limitations of the clinical trials to show similarity. The differences between the original and the biosimilar will always be low, and that's something no clinical trial can really find. Another, for me as a scientist, the most important argument against clinical trial is that clinical trials are always performed at single label doses. And this dose of a biologic is always in the plateau phase of the dose response, not in the ascending phase. And in the plateau phase, you have no sensitivity for differences in efficacy. I told you biologics are relatively safe drugs. That means that a clinical trial will always be too small to show differences in adverse effect. And another, that biologics, especially the cancer biologics, are always part of combination therapy. And it makes it impossible to find the side effects which can be 100% associated to the biosimilar. So what I'm advocating now is a new biosimilar regulation that for most candidate biosimilars, I think biosimilarity in quality, so in physical chemical characterizations, etc., will be enough. That's more or less the same approach as we have with small molecules, the generic approach. The emphasis in quality should be on the impurity and the potency. I explained to you why. And if clinical studies are needed, in my view, they will not be needed in many cases. I think a well-designed pharmacodynamic study should be sufficient. And I would only do immunogenicity study if a candidate biosimilar has a high level of aggregates. Well, I didn't discuss interchangeability and extrapolation, but I think that should be default for any biosimilar. And there is no need for specific studies to show that. Well, let's go to COVID-19. I think the whole issue of affordability of biologics, the whole issue of biosimilars and how to look at copies is very relevant, because this is what is in what we know now as biologics that have an effect on COVID-19. Of course, we have the vaccine. We have more than 160 vaccines on the way, and of some vaccines we already know that they induce an immunological response in patients. In many countries in the world, including the Netherlands, where I come from, the human antisera from recovered patients are isolated and used now in the treatment of patients. There are a number of monoclonal antibodies already available, but on the way to treat and protect COVID-19 patients. Of course, the neutralizing anti-SARS-CoV-2 antibodies. I think I counted there are at least 7 or 8 candidates on the way. And of course, we have seen studies in which immune suppressive monoclonal antibodies like IL-6 neutralizing antibodies have been used successfully in the immune suppressive regimen that is now given to patients with severe COVID-19 symptoms. And I was also delighted, because I started my career in interferon research, that interferon beta has been developed, and there is now a trial showing that it can be used in the antiviral treatment of SARS-CoV-2 infections. And of course, interferon beta will be an ideal part of a combination therapy, because my prediction will be, but it's an easy prediction, because we see that in many viral infections that the approach will be combination therapy. So what is the solution to the affordability and access problem? I think what's urgently needed is a revision of the drug regulations. And in the drug regulations, I think affordability should become a key issue, not only safety and efficacy, but also affordability. And if we look at the future of big pharma, and it looks not very good for big pharma, I think that the future should be in, we should not become too dependent on big pharma. And the only way to do that is by decentralization of drug development and production. And we see that happening in the world. This is happening in the US, where a number of hospitals are so fed up with drug pricing and drug shortages that they started their own drug company. This is an analysis what hospitals in the US do to fight drug prices and shortages. And you see that nearly 70% of the patients, the second bar from the top, is doing more in hospital compounding. That means that they are making the drugs in their own pharmacies. What we have been advocating and what we are also trying to organize on a world scale is what we call bedside production, magistral production. That means that it is produced in the hospital directly for an individual patient. And the legal situation in most of the world is that you don't need any marketing authorization if you bring a product to patients this way. So I think the future is in decentralization. We will have a global level of production. That's for the mass production of for instance, vaccines. A regional level where we will have the production and compounding of orphan drugs or limited population, and we will have production on the hospital level, and that will be for either the experimental drugs or drugs for individual patients. What's the current main issue now for the third world countries? I think we need as soon as possible a revision of the WHO biosimilar regulation, because I think it's at least the interpretation of many national authorities is that you need clinical trials in every circumstance. And that, of course, will make it nearly impossible for local producers of biologics to get their products on the market. Thank you.
Thank you, Dr. Schellekens. That was a very useful talk. I think we kind of get a basic introduction as to what we can expect and so on. And this somewhere links us with the next speaker, who would be next speaker and discussant, who will be speaking about what we can expect. I mean, is there a lot of scope here, or are we having too many expectations from the biologics in this condition? And how to look at, I might be wrong in my scientific term, but short-term immunity till the vaccine comes. Is there a role that biologics can play and so on? And I'll ask Professor Satyajit to take it over from
here. Thank you. That was an outstanding setup by Professor Schellekens. So I'm not sure that I have a great deal to add about the broader issues that he has outlined. So let me address 2 or 3 issues to which he has indicated. And let me start at the end, which is his argument about magistral approaches. One of the issues with biologics is that a very substantial proportion of them, not all biologics by any means, but a very substantial proportion of them are, and for safety's, individual patient safety's sake, can only be given in hospital circumstances. They are large proteins, they have to be infused. There are exceptions to this. Certainly the growth factors are exceptions, for example, as is insulin. But nonetheless, it remains true that a very large proportion of biologics tend to be given in hospital circumstances. That presupposes that widespread use of biologics will be associated with widespread access to hospital facilities that include the capacity to administer biologics. And I think that particularly from the point of view of accessibility of biologics, this basic limitation on the availability for the communities, especially in the global south, to hospital facilities is something that we should keep in mind, particularly from the point of view of what activism should be focused on and thinking about in terms of demands. So that's a first point. A second point that I would like to make is again related to the realities of developing economies. And let me connect this up to COVID-19 and to an example I'm going to draw from a likely scenario that will come about with respect to COVID-19 by the end of this calendar year, which is we're going to see half a dozen vaccine candidates that are going to show us data about protective efficacy reasonably soon. And I suspect, unless there are unpleasant surprises, which can never be excluded, that we will begin to see that more than one vaccine candidate of the ones that are in phase 3 clinical trials will show us some reasonable degree of protection. I doubt that any of them is going to show us absolutely spectacular levels of protection. That would be a pleasant and unusual surprise. But they're going to show us respectable protection, significant protection, one way or another. Now, I'm remembering our old friend, Dr. Amit Sengupta, and Amit's and my conversations about are vaccines to be considered part of biologics or not. And I'm not revisiting, I note that Professor Schellekens also made reference to vaccines while leaving them sitting on the edge of the discussion about biologics for perfectly good reasons, given his focus. And I'm not about to pull vaccines into the conversation about biologics. What I'm using this for is the following. If you look at all the vaccines, with the exception of the Chinese vaccines, they're all vaccines that have been developed by public sector funding in academic research institutions. And yet all of them have partnerships with, by and large, multinational biopharma, and certainly with the private sector, without exception. This is associated with payments upfront from a variety of companies, from a variety of country governments to these companies for priority bookings of vaccine doses on speculation. So the question becomes, here is an object lesson. The vaccines that are under discussion are like biologics. I'm leaving aside the question of should we include them in the conversation or not. They're sufficiently like biologics to be relevant in the sense that their manufacture depends on the kind of technologies, by and large, that Dr. Schellekens pointed out was involved in the manufacture of biologics. Is it going to be possible for governments of the global south to say, these are utterly essential, and unless we get access to vaccine doses in good time and in affordable prices, we are effectively going to start licensing them for manufacture in our own territories. And the question here becomes, is this manufacture going to be possible? Technically, practically possible. These are not easy, simple chemical synthesis technologies, which can be done by a variety of very small operators. They are demanding technologies, not simply in their establishment, but also in their maintenance. As Prof. Schellekens pointed out, batch-to-batch variation is a major issue with biologics, and batch-to-batch variation, as he pointed out, in the reference product, in the originator product, is quite substantial and significant. So maintenance of quality assurance, quality control pathways for these technologies is not simple or straightforward. So which companies in the global south will take up a challenge that governments will throw at them, even if governments summon up the political will to throw it at them, of manufacturing these vaccine candidates as actual vaccines to be distributed in the countries? And the short answer is, this is a vanishingly small slice of companies, even in India, which supplies generic vaccines across the world. I am by no means convinced that we will find local vaccine manufacturers which will take on this challenge of establishing the technologies, which they can do, but in the process, going head to head with the original IPR holders and the transnationals with which they routinely do business in manufacturing these technologies, in manufacturing these vaccines. So my question becomes, who would, under these circumstances, realistically actually manufacture vaccines against COVID-19, breaking IPR in the interests of essential need? And a huge gap, as far as I'm concerned, in all of this is the lack of public sector biopharma industry. So India's experiences with public sector biopharma industry are well known, and I'm not going to go back 40, 50 years in the history. What I remind all of us is that nowhere in the world, with again the exceptions of China and Cuba, nowhere in the world do I see public sector industry that is capable of standing up when, if governments pull together the political will and say, here, we will take on the technology, we can synthesize the genes and the vectors, we can put it all together, we can make a chimpanzee adenovirus vector-based COVID-19 vaccine that's called the Oxford vaccine, we can synthesize the necessary sequences, and we can put together the encapsulated mRNA vaccine that is the Moderna vaccine candidate, without worrying about the long-term, short-term commercial repercussions for them. The public sector industries that we had in India that were capable of doing this, we have steadily and systematically over decades starved both of the development of human resources and of the development of technological bases, with the result that I'm not sure we have this anymore, but I'm increasingly sure that we need this. And COVID-19 as a pandemic is only one example. There are going to be more, and we are going to need more of these. And the reason I give this example, and I draw COVID-19 pandemic into this conversation, is that with respect to the broader issue of biologics, I'm arguing that the one thing that we have been missing out on is demanding a public sector, industrial sector that is capable of providing biosimilars. So I'm aware that at one end of the solution spectrum is Professor Schellekens' argument about hospitals building their own companies and of magistral formulation. And I'm arguing that part and parcel of the solution spectrum also needs to be, particularly for countries in the global south, with deficiencies in the kind of hospitals that can build companies, and the kind of hospital-based resources that can do magistral dispensing. I'm arguing that we need to think about public sector industry as an absolutely critical player going forward in making biologics available widely and at reasonable, affordable prices. So that's my sort of major comment, if you will. The COVID-19 story has also brought something else forward about biologics. And that is the apparent difficulty of actually doing good clinical trials. So let me give an example. The interferon, the type 1 interferon that Professor Schellekens made reference to was shown quite some time ago as a reasonably good clinical trial to have a significant positive effect in moderate to severely ill patients of COVID-19. But that's the only trial, formal randomized clinical trial, that I have seen published on interferons. And yet, if you look at remdesivir, ritonavir, lopinavir, favipiravir, and so on and so forth, we have multiple trials. So it's interesting for us to consider this difference. Again, tocilizumab, which Professor Schellekens made reference to as the anti-interleukin-6 antibody, which sadly does not really seem to have any major effect, or the even more immunosuppressive negative signaling antibody against CD6, itolizumab, that came from Cuba via an Indian private sector, Biocon, to the Indian market. Again, we are really deficient in formal, well-done clinical trials. And it might be interesting for us to discuss why biologics in the COVID-19 sphere seem to be lagging behind in randomized clinical trials, whereas small molecule drugs, even when they have relatively little rationale or justification, I of course am referring to hydroxy, as the President of the United States of America referred to it, hydroxychloroquine, but a whole range of other small molecule drugs are being subjected to multiple repeat clinical trials, which is how it should be. Why aren't the biologics going that route, and what lesson does that hold for us? Is a question that I would ask both Professor Schellekens and many of us in the discussion group and audience. As far as I'm concerned, The issue of biologics that Professor Schellekens pointed out as a major issue is the issue of the WHO and its approach to the regulation of biosimilars. And a question that I would like to ask in this context, again, in the prevailing global political situation, is how significant is this going to be? How much traction is there going to be in all this, given the fact that the United States has given formal notice of withdrawal from the WHO? What are going to be the consequences of that purely, shall we say, petty political decision on the issues of more intermediate and long-term significance about the regulations of biologics that we are discussing? Now, really, I think that those are sort of comments and additional points to think about that I really wanted to bring up, since this is not my first time of listening to Professor Schellekens, and I was quite certain that he was going to do a very thorough and comprehensive presentation about the framework and the basic core issues involved. So I suggest that I stop here because I would dearly like discussions rather than going on about this further. And I see that there have been already 6 questions and comments made. And I also note that even in the chat box, there have been a couple. So I suggest that we begin to take those up.
Yeah. So I will kind of read out some of the questions out. And thank you so much, Satyajit. I think that understanding that of public sector also having the ability to manufacture is quite important, that sometimes we seem to be missing. So I'll read some of the questions and I'll really leave it open for either of you to address it. But I understand that some of them were addressed to Professor Schellekens at the starting. So there is a talk about inhaled interferons for treating COVID. Is there actually any possibility for use outside of hospitals in such cases, or should we be concerned about any side effects? So this is one. And the second one.
So maybe we can go one by one.
Perfect. There are just 5 questions. That would be great.
Professor Schellekens, will you take this? Well,
I will try to answer it. I think the question is whether it's possible to do these inhaled interferons in every hospital in the world. I think the data are not yet really confirmed that the inhaled beta interferon is going to work in COVID-19. And one of my comments was, I was a bit surprised that they used beta interferon instead of alpha interferon. And why they used this inhaled route of administration. And I think that has to do with the fact that you don't need so much beta interferon if you do it as a local treatment. So I think I agree that we need more clinical trials on this approach with interferon, and that should include alpha interferon, and that it should include sub-Q or intramuscular injection of the alpha interferon. Now, to answer also the question, why is there only one clinical trial? I think the answer is quite simple. both beta and alpha interferon are all drugs. So there is no exclusivity anymore. And you can also buy your alpha and beta interferon very cheaply in Cuba and in South America, and I think in India. So there is no real financial push and profit push for these clinical trials as we see with the, for instance, the Gilead antivirals. I think Dr. Rath will agree with me that that's probably the main driver of clinical trials is, as always, try to maximize profits.
Absolutely. I couldn't agree with you more. And in fact, that was the point that I was, without explicitly saying so, trying to make, that this is a major issue. But going back to the inhaled interferon, I have seen only one formally published paper on inhaled interferons, pegylated interferons. I forget if it's alpha or beta, but inhaled pegylated interferons for COVID-19, and that has not shown any differences in a case control study. I haven't actually seen a randomized controlled trial at all. One of the issues at stake may well be that the nasal and upper nasopharyngeal location for SARS-CoV-2 may not be the absolutely critical one as far as clinical progression is concerned. The clinical progression may be from much further down. And whether the pegylated interferon in COVID-19 patients does actually reach all the way down may itself be a question. But as I said, while type 1 interferon has proven to be useful in a randomized controlled trial, albeit with relatively few follow-ups or no follow-ups, the only inhaled study is a case-controlled study without a positive outcome. Can it be given in a non-hospital situation, which was the question? And potentially yes, but I would wait for an actual clinical efficacy before I even begin to think about that question.
So,
are you guys going to read the questions?
Yeah. I'll read the questions, or you can read it yourself. Would you want? The next question is by Chase. He's asking if beyond the scope of the talk, he's interested in the overlap and distinction of biologic drugs and biological products that are key components for tests detecting antibodies, antigens. And the general question would be, how does manufacturing patents regulation differ between biological products used in diagnostics and biological products which are used as drugs?
So maybe Professor Schellekens can look at how does manufacturing IP regulation differ between biological products used in diagnostics and biological products used as drugs?
I know it for Europe, but I think it will be different for the US, where everything is regulated by the FDA, and I don't know about a country like India. But I think in Europe, the drug regulations are completely different from the diagnostic regulations, where you need to get a CE mark, and that's mainly based on the technical outcome of your assay, and not on the way that the different reagents were produced. So it's more directed to the claim of being a good diagnostic than somebody really getting into details into the manufacturing process as they do with a biologics which is used as a drug, at least in Europe.
So I just got a note saying Richa is going to answer this question. Is that right?
No.
Oh.
I was just putting it off. Yeah.
Oh.
Please go ahead. Please go ahead.
Okay. Sorry. No, I misunderstood. All right. So I actually think it's an interesting question because as Prof. Schellekens says, the diagnostic test intellectual property right is a much looser concept than obviously a drug or biologic or not. And I'm afraid I don't know the answer to that question about how, whether IPR actually applies to the individual products that go into the making of a diagnostic test, particularly the biologics like monoclonals that go into the making of a diagnostic test in terms of detail. I know it can be done, but in terms of the details of the ambit of the patent, I really don't know a good answer to this. And if anybody else does, do please chime in and comment on it. But for the moment, I'm afraid that we would need to leave it there. I'm not sure that there are a lot of true overlaps between diagnostics and therapeutics. I can imagine some monoclonal antibodies being used for both, but by and large, perhaps not. So the next question is, from Brooke Baker, we could produce bioidentical if we had access to cell lines and other biological assets and to trade secret protected manufacturing processes and know-how. We rarely discuss developing mechanisms for overcoming trade secrets and mandating access to biologic resources. Shouldn't we begin to discuss mechanisms for overcoming trade secret barriers so that bioidenticals can be produced, essentially eliminating the need for more clinical trials other than bioequivalence studies? That one's for you, Professor Schellekens.
Again, that is an interesting question. I think you should not overestimate the importance of trade secrets in biologics. I've been part of the Dutch and European regulatory system. And I've seen, I think, more than 200 different dossiers on biologics. And I can assure you that I never found something in these dossiers that I did not already know. And if you look at the way that the production technology has matured, everybody is doing the same. Everybody is using the same steps of purification and fermentation. They buy their installation reagents from the same manufacturers. So I think there is more the same between manufacturers than there is different. But even saying this, even if you could provide me with the clones of AbbVie, of Humira, and give them all their technology, how they produce it. And I would start it in Rotterdam. I'm assure you that I will get a product which is different from the AbbVie product. There will be differences, but as I try to tell you, these differences are probably not clinically relevant at all. So that there are two sides to the coin.
Yeah. So simply as a follow-up comment, the one way of looking at this is that if you look at the argument that Professor Schellekens is making for a Mark II version of regulations for biosimilars, that in fact is effectively saying that biosimilars really need bioequivalent studies. and not clinical trials, without even having to force any revelations of non-existent trade secrets. So I agree with the thrust of the question, but I really do also agree with Professor Schellekens that these are not major issues. As he pointed out, in the originator product, batch-to-batch variations are quite substantial and significant. So really, we need to argue that biosimilars should be regulated in bioequivalence terms, primarily, without overstating the importance of trade secrets. The next question comes from Suhas Kolekar, a virologist. And the question is, would you elaborate on biosimilars are relatively safe? Professor Schellekens.
Again, a very good and provocative question. We, at Utrecht University, when the first biosimilars were introduced in the Netherlands, we've tested every batch of every biosimilar that came to the Dutch market, until my students were so fed up with doing the same thing over and over again that they refused to test any more batches. If we looked at the batches of the biosimilars in general, their quality was better than the originals, which is easy to understand because the biosimilars are made by the newest technology, while the original manufacturer by the regulatory systems in the world are locked in their original manufacturing process. Of course, they change it once in a while, but still they need to follow the basic concept as it was discussed in the marketing authorization. So if I would make a very provocative answer to the question, I would say that biosimilars are probably more safe than the original, but that's probably also an overstatement. So I think it's perfectly possible to make a safe biosimilar and a copy of the original.
So let me just add a footnote to that comment of Professor Schellekens. I think that the concern that alterations in the three-dimensional structure of biosimilars, either because of post-translational modifications and or because of folding alterations, are safe in the sense that apart from immunogenicity, as Professor Schellekens pointed out, they do not generate new unforeseen and potentially unsafe biological activities. And that's simply, again, as a footnote to the response that Professor Schellekens made. The next question is from Krishnaswamy Shankaran. Cuba has used interferon alpha as treatment in COVID-19, not listed in your slide. Your comments.
That may well be true. I visited Cuba many times. I know they have a relatively good alpha interferon. They also exported this alpha interferon to Iran and a number of other countries. And I wouldn't be surprised if they are testing the alpha interferon in COVID-19. And I'm looking forward, by the way, to see their results.
Yeah. And I haven't actually so far seen a trial result for that. Question from Brooke Baker. With respect to the suggestion for public sector manufacture of biologics, what is the perspective on production for export to countries with insufficient or inefficient manufacturing capacity? It's not it's a great question. It's not a straightforward question to answer, but I would argue that it falls in pretty much the same category as non-biologic drugs manufactured by public sector enterprises and undertakings. In other words, there isn't a biologic specific perspective for public sector manufacture with respect to export to countries which need it. Simply as a repeat push, my argument for including the public sector manufacture in biologics was primarily driven by the fact that in the private sector, players who would have credible technology capacity would not be willing to go head to head with IPR holders, and players who would be willing to do that may not have the capacity. This is a situation unlike small molecule generic drugs, which is why I'm introducing the argument that we as in public health activists, should be including public sector manufacture of biologics, particularly into our view of the biologics sector in territory. The next question is from Malini Aisola. Would love to hear your expert view about the Roche trial on tocilizumab and your views about the significance of this particular trial. It is obviously being discussed in terms of the effectiveness of the drug, but also for the mechanism of action. Do you feel there is much unanswered and that further trials can address? Do you want to respond to this, Professor Schellekens?
I think you're more aware of these trials than I am.
Okay, so certainly it is being discussed in terms of the effectiveness or the lack of it emerging from the clinical trial. So for what it is worth, and this is purely in my view as a T-cell immunologist, I think that what it underlines is the fact that in situations of cytokine storm, we have been for a very long time in the field thinking about it as though every patient with a clinical presentation of cytokine storm has the same cytokine storm. And yet, the one thing that dealing with patients of septic shock, endotoxic shock, cytokine storm has been teaching us repeatedly is that these are extremely diverse pathophysiologies, that there is a great deal of heterogeneity in patients that at first glance look very similar clinically. And I think that that's a fundamental difficulty that is being underlined by the fact that dexamethasone, as a very blunt hammer of the anti-inflammatory kind, clearly works to a significant extent, whereas far more specific single cytokine therapies may not quite work, simply because the patient group is so heterogeneous that we are missing the patients in whom there is a response because of the heterogeneity in the design of the drug. But that's really just a speculative view.
So in the chat, we have a question actually, which says, what would you recommend to support small enterprises research and development for MABs, as big pharma is ahead in the race, but not necessarily that their monoclonal antibodies will be the most potent or the cheaper ones? This is in the chat box.
I'm happy to answer that. Professor Schellekens, would you like to comment?
No, answer it, please.
Okay, so So let me take an emboldening page from Professor Schellekens' book and make a provocative argument here. Do I think that MABs, monoclonal antibodies, are worth? Did I understand the question correctly? I'm scrolling to see if I can find it.
It's in the chat box if you would like to see it.
Is it worth for us to for small R&D establishments to make them? Given
No, I'll read it again. What would you recommend to support small?
Absolutely. So here's the provocative version of that argument. Do I think that it is worthwhile? Absolutely, yes. Why do I think it is worthwhile? So I think it is worthwhile for three different reasons. Number one, as Professor Schellekens pointed out, monoclonal antibodies in the COVID-19 context can be divided into two separate components. One is monoclonals that are neutralizing antibodies for SARS-CoV-2, for the virus. And we need monoclonals that neutralize, because different monoclonals will have different potency, number one, because their binding abilities will be quite different. The biophysics of their binding will be quite different, and that will have an effect on how efficiently they will block the virus virus particles from infecting new cells. Secondly, although SARS-CoV-2 as a coronavirus and as a very large genome coronavirus is not as heavily mutating as many other RNA viruses are, it is an RNA virus, it will mutate, it will be subject to immune selection, we will get variations. We already seem to maybe have gotten a variation at position 612, but we're certainly going to get more variations. And therefore, if SARS-CoV-2 neutralizing MABs are going to be an actual product going forward, there is space for many more products in the neutralization field. On the cytokine-directed MAB front, again, as we just discussed with tocilizumab, I don't think that any one cytokine, even in the inflammatory cytokine kit bag, is going to be really successful and useful for all patients suffering from severe cytokine storms. I think combinations are going to be important. I think multiple different combinations are going to be important going forward. There are already emerging data that there are at least subacute, if not chronic sequelae to severe COVID-19, and the inflammatory landscape there is going to be yet another region where, given the large numbers of patients, there is going to be a need for MAB kind of therapies. So that's my second reason. But my third reason for saying that everybody needs to be working on these innovative products and technologies is simply the argument that we need to make the case credibly that none of these are true designed innovations. They are simply empirically discovered entities. And even more so from the point of view of either vaccines or SARS-CoV-2 receptor binding domain directed or cytokine directed MABs. That's prior art. We already know that we can go on making MABs against these targets, and many of them will work. But in order to make this case for the non-innovativeness of these products, in contesting intellectual property rights being claimed, we need actual practical support. And that part of that support, I suggest, is going to come from a very wide, across the global south, public and the global north, publicly funded small research groups coming up saying, hey, we made this, we made another, somebody else made another, somebody else made another. That's the kind of empirical evidence that we need to gather together in support of the claim that a lot of this does not involve a truly innovative step. So that's my combined argument for that particular.
So there are questions which are coming in, but we might have to wind up in 10 minutes or 5 minutes, because I think Professor Schellekens also needs to go. I would maybe, if you can skim through the question, and if you can skim through the question and also give your parting comment on the issue, the topic, that would be a great to end it.
So, Professor Schellekens, there is a question from Santosh saying, compared to the US, there seem to be more biosimilar approvals in Europe. Does this mean the EU is approving biosimilars without sufficient data?
Professor, please unmute
yourself, sir.
Oh yes, I have to unmute. The point is that we were the first in the world with biosimilar regulations already in 2006, while in the US, I think the first regulation came in 2014 or something. So it's a reflection of the length of time we have the regulations. Saying that, I think also everybody would agree in this moment in time, the regulations in the US are probably more demanding than in Europe, where we are now more relaxed on biosimilars than 15 years ago.
So let me
mean that the biosimilars are bad in Europe. We are only more relaxed about the biosimilars at the moment.
So let me press quickly the next question onto you as well. Is there good data on the evolution of costs of producing monoclonal antibodies? Has it decreased many times over the past decade or so?
Yes. We have data that about, let's say, 10 years ago, the cost of producing 1 gram of monoclonal antibody, which is a lot, by the way. You can treat a lot of patients with 1 gram with most monoclonal antibodies. Was about $300 to $400. By now, it's probably 5 to 10 times cheaper.
Yeah. Okay. The last question is a long and overarching one, so I'm going to read it out, and I'm going to ask Professor Schellekens to both respond to it and to give his final comments. The overwhelming impression from KB, the overwhelming impression from research reports is that trials and research seem to be largely competitive and solo acts by companies, some supported by governments. Is this true, or is there a real effective collaboration taking place among researchers and scientists behind the scenes? If not, is there scope for setting up platforms outside of corporate structures to promote collaborative rather than competitive research for COVID-19? And do we have good past examples of such collaborations? Your comments on that, Professor Schellekens, and your final wrap-up comments, both.
Again, it's a matter of who's behind the research. If it is research driven by trying to get a product on the market as soon as possible by companies, then there is a lot of competition. But luckily, still in microbiology, there is still between academic institutions and also coordinated by WHO or by COVE, the central vaccine operation. There's still a lot of collaboration going on. But again, if there is no commercial goal, then everybody is collaborating happily. If there is a commercial goal, then we live in a different world than 30 years ago in microbiology.
So simply as my wrap up, because this brings me back very smoothly to my core point again. Are there true collaborations, as Professor Schellekens points out, so long as it's about understanding COVID-19? There are transnational collaborations of the academic kind built across the world. Are there drugs, vaccines related technology-based true collaborations? And the only ones that I'm really aware of are, again, the ones that Professor Schellekens pointed out, Gavi, CEPI. These are multilateral, broadly philanthropic collaborations that pull philanthropic charities, some nation-state governments, and the biopharma sector together in a very awkward bundling of the public good and the profit margin. And I am arguing, once again, as simply my wrap, that we will not be able to build a true non-corporate sector collaborative effort to build solutions if for the last mile of making the products, we don't have public sector industry. In the absence of that, ain't going to happen. So, as I said, quite conveniently for me, that also is my closing comment wrap up. So back to the hosts.
Yeah. Thank you so much. I think the last long question is definitely from Kajal, if I can understand. And she has been part of those people who have been very angry with whatever is happening in the patent and access to medicines landscape. But I think at the end of the whole session, we kind of also come back to the same issues about R&D and manufacturing, which we have been seeing from quite some time, and hope that and as PHM and as TWN and various organizations, we hope to put our efforts in the way which also not just are looking at our short-term goals of immediately, but as you are saying, push or advocate and conduct our activism, which means for a public policy change where public institutions are strengthened. I think and also there is a very kind comment in the chat box from Carolina Gomez that you have a lot of fans in Colombia.
I was about to say that in wrap, I thank all the audience, including old friends from Colombia, like Carolina Gomez.
Yes. So thank you so much, Professor Schellekens, and thank you so much, Satyajit. It has been a pleasure to have you in the webinar, and we'll be uploading these videos and circulating it to everyone to look at it. Thank you so much again.
So just one point, there is a question in the chat box about whether Professor Schellekens would share his slide set.
If it is fine, I can upload it on the PHM website. Sure.
Because if it's okay, then they can share it.
Yeah, no
sure.
problem.
We'll upload it on the PHM website and we'll share it across on our mailing groups and everything. Thank you so much. Thank you.
Thank you.
Thank you and good night.